A very good day to all of you. Today we will be discussing on the physiology of reproductive system. In this lecture, we will discuss on the physiology of male reproductive system. At the end of this lecture, you should be able to describe the changes associated with puberty in males and females. You will be summarizing the formation of gametes with reference to spermatogenesis. We'll be describing the spermatogenesis and its regulation by hypothalamic-pituitary-testicular axis. We'll be also describing the actions and regulation of testosterone. Reproductive system does not have any role to maintain the homeostasis. This is the first and foremost thing you need to understand when compared to any other systems in physiology. There are many systems like renal physiology, which is dealing with the ECF volume regulation, the nervous system, the endocrine system. Everything is contributing to the homeostasis of the body. But in the case of reproductive system, it does not have any role in the contribution to the homeostasis. The individual can very well survive without a reproductive system. Even if the reproductive system is removed, he can still survive. He or she can still survive without a reproductive system. It is essential for the production of the next generation and species survival. So that is the most important part. In the reproductive system, the only thing it is playing is it is having a very important role in the production of gametes, so that it will be essential for the species survival. But it does not have any role, or it does not contribute anything to the body homeostasis. Primary sex organ or gonads. In the case of males, it is testis, and in the case of females, it is ovary. They help in the gametogenesis, means the production of gametes, and also for the secretion of various sex hormones. Gonads produce sex cells called gametes and also secretes the sex hormones. There are accessory reproductive organs, like in the case of ducts, glands, and external genitalia, which does not contribute directly for the production of gametes, but they aids the process of fertilization, the process of reproduction. So that is the only thing, but otherwise it doesn't have any role in the formation of gametes. That's why they are called as accessory reproductive organs, like ducts, glands, and external genitalia. Puberty or adolescence. It is driven by the maturation of the brain. Increased secretion of hormones from the hypothalamus and pituitary gland is responsible for the puberty or adolescence. Growth and development of gonads or sex organs will be taking place at the time of puberty. There will be development of secondary sexual characteristics in the individual. Generally, it occurs between 8 to 13 years in the case of girls and 9 to 14 years in the case of boys. So here we need to remember that the girls mature faster than the boys, 8 to 13 and 9 to 14. So we'll come to that part when we discuss about the secondary sexual characters and other things later in the next slides. During puberty, both genders experience great mental and behavioral changes. Definitely when the individual from the adolescent period, when they attain maturity or puberty, there will be definitely a mental and physical changes or behavioral changes is clearly evident in every individual. Hormonal changes during puberty. The hypothalamus synthesizes and secretes gonadotropin releasing hormone, which is called as GnRH, that is produced from the hypothalamus. GnRH acts on the anterior pituitary, and on the anterior pituitary, this particular hormone GnRH will be stimulating the synthesis and secretion of follicle stimulating hormone, which is called as FSH, and luteinizing hormone, which is called LH. So what it is doing, hypothalamus will be secreting gonadotropin releasing hormone, which is called as GnRH, which will be then acting upon the anterior pituitary to synthesize and secrete the hormone FSH, or follicle stimulating hormone, and luteinizing hormone. LH and FSH will be then activating the gonads, which includes ovaries in the case of females and testes in the case of males. Hormonogenesis, which means it is the production of the sex hormones, and gametogenesis means it is the production of gametes, sperms in the case of males and ovum in the case of females. Now let us see some of the important terminologies associated with puberty. Telarch, which means development of breast at the time of puberty in the case of females. Pubarch, which means development of axillary and pubic hair at the time of puberty. Menarche, the first menstrual period in females. Adrenarche, which means increased secretion of adrenal androgens at the time of puberty. Secondary sexual characteristics are differentiated, like in the case of males and females. You can see that in the case of males there are several features, in the case of females also there are several features. Increase in the height, definitely you will be seeing that there is increase in height in the case of males and females. But one thing you need to understand or differentiate, that in the case of females they mature faster than in the case of males. At least 1 year before the females will be maturing faster. So a simple example, if you see that usually at the time of puberty they will increase their height, they will become more taller. So suppose if you have a cousin sister of your same age group, and you might see that during childhood days at one particular stage when they are matured, they might be taller than you. They are taller than you because they have attained the maturity at a faster rate when compared to you, and they grow taller. Right? So that is the difference what you see when you compare to a boy and the girl of the same age group. Usually in the same age group, before just the time of puberty, what is seen is that the girls will be achieving taller or achieving better heights when compared to that of the boys. But later they will be, when the boys become matured, they will pick up their growth and then they will grow taller than the females. So that is how it is done. So here the most striking feature what I want you to realize is that between males and females, usually the females will be attaining maturity faster than males. Now in the case of males also, you can see that there are so many features: broader shoulders, enlargement of penis, scrotum, testis, formation of sperms, longer and heavier bones. So these things you need to memorize how it is. So maybe 1 or 2 features can be asked. Another striking feature what you see in the case of males is a deeper voice. That means the males, they will be having a bass voice when they turn like puberty into puberty. So this is because the vocal cords will be changing due to the accumulation of the testosterone or the action of the testosterone. The nature of the vocal cords will change and the sound will be changing, right? So that is why in the small boys, if you see that if you listen to them, it will be like a feminish voice in the beginning, but when they turn like into a mature individual, you'll be seeing that the voice changed into a bass voice, which is a man's voice, right? So that's deeper voice. But in the case of females, it is a very shrill voice, like a high pitch voice. So those kind of differences you will be seeing. And in the case of females, the beginning of the menstrual cycle will be one of the important feature what is seen in the case of puberty. Increase in breast size, formation of mature ova. So all these features you need to differentiate that from that of male. Male reproductive system. Gonads produce the sex cells called gametes, and also they are responsible for the secretion of the sex hormones. There are accessory reproductive organs such as ducts, glands, and external genitalia that facilitates the reproductive process, but it does not have a direct role in the production of gametes. The sex hormones, androgens, are seen in the case of males, and estrogen and progesterone, they are seen in the case of females. So in this picture you can see that there are various organs which is mentioned. This is the primary sex organ, which is the testis. Then we have other structures like epididymis and other structures which are accessory, which are part of the accessory reproductive organ, and also the external genitalia. So those parts other than the one which is helping in the reproductive process, that is called the accessory reproductive organs, which does not contribute in the production of gametes. So in this picture you can appreciate various anatomical structures involved in the male reproductive system. So I request you once again to go back to your anatomy slides, lectures, and have a review of those structures carefully before you look into the physiology part. Spermatogenesis, it is the production of gametes in the case of males. That's the male version of gametogenesis. They occur within the seminiferous tubules of testis. It is highly depending upon the optimal conditions for the process to occur correctly, and it is very essential for the sexual reproduction. Starts at puberty and usually continues until death of the individual. So this is one striking feature when compared to that of a female. In the case of a male, male's reproductive capacity right from the puberty till the death, it is perceived. That is why even if the individual is old, an old man can still become a father, because his reproductive capacity has no limit. Of course, there are, as age advances, the level of testosterone and other things might reduce, but still he is still capable of becoming a father, right? So that is the one difference. But in the case of females, after menopause, they will not be able to sexually reproduce. So that is one striking difference what you have to differentiate between the two genders. However, the production of sperm decreases with increasing in age. Spermatogenesis is the production of spermatozoa from the spermatogonial stem cells by mitosis and meiosis. Initial spermatogonia yield primary spermatocytes by the process of mitosis. Primary spermatocytes then divides by first meiosis into two secondary spermatocytes. Each secondary spermatocyte then divides into two spermatids by the second meiosis. Spermatids develop into mature spermatozoa, which we call it as the sperm cells. Thus, a primary spermatocyte will give rise to two secondary spermatocytes, which by their division produces 4 spermatozoa. Spermatozoa are the mature male gametes. Mature male gametes, they are called as spermatozoa. So in this picture also you can see that this is the spermatogonial cells which is having diploid number 2n. It is diploid in number, the chromosomal pattern, when after the first spermatocyte, primary spermatocyte, then it forms, after the first meiosis, it forms the primary spermatocyte. Again, it is diploid in number. Then the second meiosis, at the second meiosis, you will be seeing that when it is dividing into two cells, which is called the secondary spermatocyte, which is haploid in number. So the chromosomal number from 2n, it is reduced to n. So it is the chromosomal number is halved. After that, the four cells which is formed, they are called as spermatids. The spermatids, when they mature, they form into mature sperm cells, which is also called as a spermatozoa. So here, when you compare from this cell and when you reach the final mature spermatozoa, you can see that the shape of the cell is changed. From here, it was a spherical shape, but when it reaches a mature spermatozoa, it is an elongated structure with a definite head, body, and a tail. So the shape also changes. Let us now look into various features seen during the spermatogenesis. You will be seeing that there is a reduction in the cell size. The cell size is totally reduced when it becomes mature. There is also a reduction in the amount of cytoplasm from the spermatogonial cells. When you look at the cytoplasm in the mature sperm, the cytoplasm is only very less, it is reduced. Reduction in the chromosomal number from a diploid number, it is transformed into a haploid number. There is a change in the shape, definite change in the shape. From a spherical shape, it has gone into an elongated structure with a head, body, and a tail. Spermiogenesis, spermatids to sperm, that process is called as a spermiogenesis. Spermiogenesis is a transformation of the non-motile spermatids to motile spermatozoa. So spermatozoa is capable of moving, whereas spermatids are incapable of moving. Late in the spermatogenesis, the spermatids are haploid but non-motile. The spermatids will lose the cytoplasm and form a tail, becoming a sperm. So this is the features seen within the seminiferous tubule. In this picture, you can see that this particular cross section is a section of a seminiferous tubule. If you are looking from above, you can see that this is a spherical structure, and on the sides you can see that the various stages from the outermost circle, that will be the spermatogonial cells, and when you're seeing that, the matured cells will be more seen, and finally the last layer, what you see here, that you are seeing as the tails of the mature sperms. So mature sperms will be just hanging around from the interior part. So you will be seeing that various stages of the spermatogonial cells will be there, and finally you will be seeing the mature cells in the centermost region. So that is the cross section of a seminiferous tubule, and also it will be remaining attached to a special type of cell which are called as the Sertoli cells. So the cytoplasmic folds of the Sertoli cells will be helping to this attachment. So this Sertoli cells is one of the supporting cells and the cytoplasmic folds of the Sertoli cells onto which the sperms will be getting attached, and then maturation will be happening there. And here it is just given the various stages: spermatogonium, then it is forming primary spermatocyte, secondary spermatocytes, spermatids, then spermatids undergoing packaging, and then mature sperm. So that's the various stages will be appearing. So when you're looking from here, the outermost part in the seminiferous tubule, that will be consisting of immature cells, which are spermatogonial cells, which are the stem cells. From that you will be seeing that various maturation will be going on, and finally in the centermost part you will be seeing the tails of the mature sperms. This is once again the histology of testis, which clearly shows a cross section of the seminiferous tubules. So in this picture also you can appreciate that the outermost regions, this is the outermost region, these are the spermatogonial cells. We can see here, see spermatogonial cell, that is the outermost, the dark colored cells, what you see in the outermost region, they are the spermatogonial cells. Then you will see all these developing cells, different stages you are seeing, and finally you are seeing the spermatozoa, which are the mature sperms, and these are the tails of the spermatozoa, what you are seeing as elongated structures, right? So here it is mentioned varying stages of the sperm development. So all these are different stages, and finally this one you are seeing here in the centermost area, that is called as the spermatozoa, which are the mature cell. Another cell you need to carefully observe is the Leydig cells. Leydig cells is not seen inside the seminiferous tubule. It is outside the seminiferous tubule. So every point where you see outside, it is the Leydig cells which are responsible for the secretion of testosterone. So testosterone is actually produced by the Leydig cells, and then testosterone will be diffusing inside the seminiferous tubule. Once it is produced outside, it will be diffusing into the seminiferous tubules. So this is one thing you need to be careful and try to understand that. So Leydig cells, or the testosterone cell producing cells, is not found within the seminiferous tubule. It is outside the seminiferous tubules, and then the testosterone will be crossing this and then entering into the interiormost region. So we'll discuss about that later. Sertoli cells or sustentacular cells, and we will see the functions. Sertoli cells, they function as the blood-testis barrier. What is the blood-testis barrier? That means just like the blood-brain barrier, what we have learned before, a blood-testis barrier means the blood will not get mixed with the testicular contents. It will not be entering into the seminiferous tubules, and the sperms are again a highly sensitive structure which has to be kept isolated. If any substances in the blood that can alter the behavior of the sperms. So that is the reason that there are so many chemicals and various changes in the blood that can alter the behavior and the function of the sperms. So it is highly isolated structure, so the blood will not be entering into the testis. And this particular cell, Sertoli cells, will be providing as the blood-testis barrier. It will also nourish the developing sperms because it is rich in glycogen. These cells, the Sertoli cells, are rich in glycogen, so which will be acting as a source of nourishment for the developing sperms. It secretes a special protein which is called as androgen binding protein. Androgen binding protein. So when it is produced, this androgen binding proteins will be seen within the seminiferous tubules, and I mentioned that the Leydig cells are outside the seminiferous tubules, which produces the testosterone. Once the Leydig cells produces the testosterone, testosterone will be entering into the seminiferous tubules, and then it will bind to the androgen binding protein, so that the concentration of testosterone will be maintained constantly within the seminiferous tubule. Otherwise, what will happen? Because the testosterone is coming from outside, from the Leydig cells, and if you don't have a source of concentration of the testosterone inside, it can affect the developmental stages of the sperm. Because testosterone is an essential hormone for the development of the various phases of the spermatogenesis. It secretes another hormone, inhibins. Inhibins is another hormone. It is not inhibit, it is inhibins. Inhibins is the name of the hormone, inhibins, which regulates the gametogenesis. They are also phagocytic in function, that means they phagocytize abnormal sperms and also the cytoplasm from the spermatids. The excess of cytoplasm also will be engulfed by the Sertoli cells. final development of the sperms will be taking place within the cytoplasmic folds of the Sertoli cells. In the previous slide, we have seen that particular picture where the cytoplasmic folds of the Sertoli cells will be holding the sperms, so that maturation of the sperms will be taking place at that particular point. So here in this particular slide, it is focusing on the Sertoli cells or sustentacular cells providing the blood testis barrier. So if you look at these light blue colored cells, these are the Sertoli cells, and into this it will be forming a tight junction so that it will be acting as a blood testis barrier, and into the cytoplasmic folds you will be seeing that the maturing sperms will be hanging around. So that will be happening. So here also you can see that this is Sertoli cell, and these are the spermatids, which will be late spermatids, which are in the maturing form. So that will be, and each of these are the spermatogonial cells. So this will be the most immature, this will be slightly mature, this will be slightly mature, like this. So the maturation will be going from this direction, right? And finally you will be seeing the sperms. So that is how it will be. Let us now look at the factors regulating spermatogenesis. The first and foremost factor is the testosterone, the male sex hormone, which is essential for the initial mitotic division and the final maturation of the spermatids. As I mentioned before also, the testosterone is released by the special type of cells which are called as Leydig cells, which are located outside the seminiferous tubules. Once it produces the testosterone diffuses into the seminiferous tubule, and then it binds to the androgen binding proteins where the concentration of the testosterone is maintained. And that is essential for the developing stages of the spermatogenesis. FSH acts on the Sertoli cells. FSH is the hormone which is released from the anterior pituitary, and that will be acting upon the Sertoli cells, and it releases a hormone called inhibins. So inhibins are produced by Sertoli cells. Then comes the luteinizing hormone, which is called LH, which will be acting upon the Leydig cells. So LH is also secreted from the anterior pituitary, but it acts on the Leydig cell. So L acts on L. If you see the first alphabet, L is acting on L. That is how you will be able to remember it. So Leydig cells will be stimulated by the luteinizing hormone secreted from the anterior pituitary. Then comes another important factor, which is the temperature. Inside the scrotum, the testicles are maintained at 32 degrees Celsius, which means it is about 5 degrees lesser than the body temperature. When the body temperature is 37 degrees, at that particular temperature, the seminiferous tubules will get degenerated if it is kept closer to the body. That is why usually in the case of males, what we see is the testicles are placed within the scrotum, and it is bit far away from the body. It is just hanging. That means it is quite far from the body. So it will not be maintaining the same body temperature. It is cooler than the body temperature. It is 32 degrees Celsius. There are certain cooling mechanisms which is there in the scrotum. So it is a kind of a counter current mechanism which is seen. You will be learning it later in detail when you further learn about the reproductive physiology. But at the moment, what you need to remember is the temperature is one of the important factor. In case if the temperature is not maintained, then the seminiferous tubules will undergo degeneration, right? If it is same body temperature, it will be undergoing degeneration. So that is what you see in some of the conditions like cryptorchidism. Cryptorchidism is a condition of undescended testis. The testicles still remain inside the abdomen during the development. It will not during the developing stages. It is usually seen inside the abdomen, and before the childbirth you will be seeing that in the case of male child the testicles would be coming to the scrotal sac. So that is the one which is happening, but in some individuals, if it doesn't cross or if it fail to descend down, an undescended testis is called as a cryptorchidism. So those individuals, if it is not surgically treated, they might undergo infertility. They will be a victim of infertility. Actions of testosterone can be classified under different categories. Now let us see the actions of testosterone during fetal life. It is responsible for the development of male internal and external genitalia, the male pattern of internal genitalia and the male pattern of the external genitalia. It is required for the development of male brain. Male brain means it is the male pattern of hypothalamic hormone secretion. In the case of a male pattern means if you look at the hormone secretion, in the case of males, it is a continuous secretion, continuous production. The gamete production is never ending. Right from the puberty till the death of the individual, it is continuous. But in the case of females, it is cyclical. Only once in a month it is happening. So there is a cyclical pattern of hypothalamic release in the case of a female, but in the case of a male, it is the kind of a male pattern. That is why it is mentioned as a development of male brain. Here the male brain specifically means the hypothalamus, the male type of hypothalamic hormone secretion. The descent of the testis, it requires the testosterone is essential for the descent of the testis. That's what I was telling. Right from the abdomen, it has to move into the scrotal sac. So that is called as a descent of testis, which also requires the role of testosterone. Actions of testosterone during puberty. It is required for the spermatogenesis to go on. Growth of primary sex organ at the time of puberty. The size of the primary sex organ increases at the time of puberty, which is mediated by testosterone. Growth of accessory sex organs. We have seen the various like ducts, glands, and etc., which are not having a role in formation of gametes, but it is required for the reproduction. So those structures also increases in size. Development of secondary sexual characteristics will be seen at the time of puberty as a result of testosterone function. Anabolic actions of testosterone. Testosterone also increases the anabolic actions like increase in muscle mass and etc. Or it is considered to be an anabolic hormone. Inhibition of luteinizing hormone secretion. So it is a kind of hormonal negative feedback which will be inhibiting the luteinizing hormone secretion from the anterior pituitary. So these are the functions of testosterone during puberty. Now let us see the action of testosterone in adult. Spermatogenesis. Because in the adult also the spermatogenesis is continuing. Maintenance of accessory sex organs is seen. This slide shows the regulation of testosterone through hypothalamic-pituitary-testicular axis. This is a very important slide to know the feedback regulation. You can see that from the hypothalamus the hormone GnRH is secreted, and that will be stimulating the anterior pituitary to synthesize the hormone LH and FSH. And LH will be acting on the Leydig cells, L and L, Leydig cells. LH will be acting upon the Leydig cells, and FSH will be acting upon the Leydig cells. The Leydig cells are the one which is responsible for the secretion of testosterone, right? So testosterone will be produced and it will be having the anabolic effect, and you can see that the testosterone will be inhibiting the LH, right? It will be inhibiting the LH, and also it will be inhibiting the GnRH. So this is a negative feedback regulation. Now testosterone also will be stimulating, you can see that it will be stimulating the Sertoli cells also. Then from FSH it will be activating the Sertoli cells, and from the Sertoli cells the hormone inhibins is released, and inhibins will be inhibiting the anterior pituitary like this. So that is a feedback regulation, a negative feedback regulation. So this is how the levels of the testosterone and the inhibins are maintained within the blood. and that is why that any time when the testosterone levels or the inhibin levels is altered, that can then by the feedback mechanism it can bring it back towards normal. So that is essential. So any damage to the Sertoli cells, what will happen if there is any damage to the Sertoli cells? It can lead to infertility, because Sertoli cells are one of the important cell which is acting as a blood testis barrier. So if there is any injury to the Sertoli cells, it can also be one of the cause for infertility. So it is not always the problem with the testosterone, it can be a problem with the Sertoli cells also. Hormones of brain testicular axis. So it's a similar picture. So in this picture you can appreciate that androgen binding proteins is mentioned. This is within the seminiferous tubules. We can see that the testosterone will be produced by the Leydig cells or interstitial cells of Leydig, which is outside the Sertoli cells, outside the seminiferous tubules. And then what will happen is testosterone will start diffusing into the seminiferous tubule, and then it will be binding to the androgen binding protein. So androgen binding protein will make sure that it is getting concentrated, the testosterone is getting concentrated within this particular seminiferous tubules. So controlled by GnRH from hypothalamus, begins to secrete FSH and luteinizing hormone. LH act on the interstitial cells of Leydig to secrete testosterone. FSH acts on the Sertoli cells. Androgen binding protein concentrates the androgen for spermatid. So this is what this particular protein is doing. So inside, because testosterone is produced from the outside source, when it enters inside the seminiferous tubules, we need to maintain a steady concentration of the testosterone. That is why androgen binding protein will be doing. And androgen binding protein is secreted by the cell which is the Sertoli cells. Inhibin will be exerting a negative feedback to the pituitary and hypothalamus. So these are all regulatory hormones. That's all about this particular chapter, and thank you for listening.